I'm Lindy. I'm going to talk about the distribution of fitness effects. I'm down here because my slides don't appear up there.
Yeah.
Thank you. Okay.
No worries.
So I'm going to go really fast. Now, the distribution of fitness effects, we all know what this is. Probability distribution of the fitness effect of rare mutations, whether they are beneficial or deleterious. We compute the fitness effect in the usual way. And my interest in this was inspired by some puzzling experimental results from my collaborator, Deepak Gashi. And as we were puzzling through them, we realized what we needed was a null model of how the DFE would change over the course of adaptation, and we didn't have such a null model. There's actually very little work in that area. The dynamic changes as adaptation proceeds. How do we expect the DFE to change its shape? We have this intuition, of course, that it's going to shift to the left as beneficial mutations get depleted by adaptation, but exactly how? Can we describe it mathematically? So to do this, we did the simplest possible model, of course. So I'm going to show you a really simple fitness landscape. It has a number n of fitness components across the top, and each component makes a contribution to fitness, these little numbers. And in this case, for the results I'll show you today, we're going to just use the average of all the fitness contributions to get the organismal fitness, capital W. And then if a mutation occurs, one of the fitness contributions will change, the fitness will change, and again, we can compute the fitness effect of that mutation in the usual way. I've also done the multiplicative landscape, and the results are the same, even stronger. And our next step is to do epistasis, because the important thing about these fitness components is at this point, they all contribute independently to fitness. And I also wanted to point out that although in this diagram we've used nucleotides, I think actually this model is better understood for larger level fitness components that we might think of being independent, like maybe, mate finding, reproduction, survival, et cetera. They could be very large scale fitness components. And then we have also the simplest possible model we could come up with for adaptation, which is an adaptive walk. And here I want to show all the work for this adaptive walk has been done by Abby, who's going to speak later today, and Tenoch. Abby did all the simulations, and Tenoch did the analytical work, and they come together beautifully. I'll show you shortly. So in an adaptive walk, we assume that the population is genetically homogeneous, and mutations are rare, and when a mutation happens, usually it's proposed, and it doesn't fix in the population, so we go back to this focal genome or consensus genome. But if it fixes with probability pi, then the genome changes, and a step in the walk is taken. And some of the notation, n components of fitness, organismal fitness, capital W, and I'm going to use lowercase w and w prime for the fitness contributions of the allele that is in the focal genome and then the replacing allele, respectively. And Tenoch's key insight, one of a couple key insights that allowed us to plow ahead with this problem, he had to figure out what we needed to keep track of and what we didn't need to keep track of in this problem. And he found out that there were 4 key distributions that we need to keep track of, and those are these L's and M's here. So Li is the distribution of, it's the probability distribution of the fitness contributions, those little numbers in the previous slide, for all of the alleles that are currently in the focal genome. M is all the available mutations, all the way across, not just those 3, all the available mutations across the whole genome. And then when a fixation occurs, we have L, the allele that got replaced at the fixation event, and M, the mutation that did the replacing at the fixation event. And I'll remind you of those again as we need them, if I have time to go through the math. So trivially, from the definition of fitness in this additive model, this is how the fitness effect looks, and we can rearrange that such that if I told you I had a fitness effect W, you could tell me which W prime I would need to replace it with in order to get a given S. And that would look like this. And that allows us to compute the DFE. So the probability that we get a fitness effect of size S, we have to have an existing allele of size W, and then we need to replace it with exactly the right W prime to give us S. So that integral gives us the DFE. And it depends on the distribution of existing alleles and available mutations. And I'm sorry, I haven't told you this subscript. This is the 0th step of the walk. So for the 0th step of the walk, these are initial conditions. We have to make up some probability distributions for the alleles and the mutations. And then our goal is to figure out the first step of the walk, the second step of the walk. What does the DFE look like? So it's actually pretty easy to figure out the probability distribution for the replaced allele, the one that gets replaced. It would start with what's the probability that it had size W, what's the probability that the mutation has size W prime, and here's that fixation probability, W prime given W. And likewise, we can do the M, the allele that did the replacing. I won't show you that. And then this was another key insight that Tenoch had. He said if we look at all alleles in the genome initially, one of them, after we make the step, is going to be drawn from the allele that got replaced, and n minus one of them are alleles that didn't get replaced, the unmutated alleles. And this U is not only unmutated alleles, it's unknown. We don't know what this probability distribution is. But we've just figured this one out, and that's an initial condition, so we can solve for it, and we do. And then we can use the same logic. The alleles after one adaptive step in the genome, one of them is an allele that fixed, that did the replacing, and we know that one. And now we know this, the n minus one unmutated alleles. So now we have L1. And again, similar logic, I won't show it to you, gives us M1. And if you remember the DFE expression, DFE0 depended on L0 and M0. The DFE after one step depends on L1 and M1, and we've got the whole thing. So there are a few tiny approximations in here, but basically Tenoch cracked it. And look at this, you can't even tell, but the bars are Abby's independently run simulations, and the solid lines are Tenoch's analytical predictions. So you do see the effects of some of our approximations here, but those are the 4 key distributions, and aren't they gorgeous? And here's how the DFE changes. If we start with an initially uniform, which is admittedly not very biological, uniform distribution for fitness effects, we get this crazy triangle for the DFE. But as time progresses and more mutations accrue, we start to approach something that's way more biological. So we have this tail of rare beneficial mutations, and then the bulk of the distribution is slightly deleterious. And Troy Day could tell you all about how we expect these all shapes to all look the same anyway. Okay, I'm doing great for time. Sorry, I'm being really fast. So this, though, is the interesting slide. So I'm going to spend some time here, and it's hard to talk through, so bear with me. So all of the results on this slide are after 50 or more steps in the adaptive walk. So adaptation has gone quite a long way. At the top, we have the DFEs. And again, we see that shape. In all the cases here, we have these rare beneficial mutations. And on the bottom, we have the distributions of the fitness contributions. So 2 of the key distributions, the L and the M. So the alleles currently in the genome are in blue, and the available mutations are in red. Okay, and what I find interesting, I'm curious to know if other people find this interesting and counterintuitive. Maybe you'll be like, yeah, that's totally obvious. To me, it was counterintuitive when I see these results here. Okay, this is again the analytical and the simulations, and we understand why the approximations don't work when the number of alleles per site is very small, so that's not the point of the slide. But here, if I were to explain why are the beneficial mutations rare at this point, I would explain it by saying, look, here are the available mutations in red, and we've run out of available mutations that are high fitness. When we run out of high fitness available mutations, the available substitutions, I'll call them, the S values are small. That made perfect sense to me, and it's wrong. I mean, it's right for a biallelic locus. So this panel, at every locus, there are only 2 options, right? But if we move to 4, for example, nucleotides, 20 amino acids, as we move up, that explanation becomes increasingly wrong. And you'll notice even here, we did an infinite alleles model, even when there are infinite alleles available, so there's no depletion of available mutations. There are always many available mutations available at each site. We still see this depletion in the DFE. So why is the DFE getting depleted? It's getting depleted because we are running out of bad things to fix in the genome. So it's not like we're running out of good solutions. There's lots of good solutions out there. In the infinite alleles model, there are infinite good solutions out there. But your genome is running out of bad alleles to be replaced. And I think why I'm interested in this is that I think that's not particularly model specific. I think in general, if you have a bunch of fitness components and the organism has more than one way to solve a problem, many ways to solve a problem, and I think that's true generally. If there are many ways to solve the problem, adaptation is slowing down, not so much because we're running out of new beneficial mutations, but because we're running out of bad things in the genome that need fixing. Okay, so that is the one exciting thing that we have, as well as like, I'm super excited that we have this mathematical description of how the DFE changes. These are my collaborators. Deep and Myrtala inspired the whole thing. There's my group, Abby and Tenoch. I'm grateful to NSERC for funding. David and Ryan also talked to me a lot about this project as it was going on, and I welcome your questions.
Okay, hello everybody. So my name is Christian Huber. I'm from Penn State University. I want to start with a statement. I'm not sure if that's very controversial, but a lot of the PopGen models of selection that we kind of use to understand what's the relationship between selection and genetic diversity in the population are based on models that assume very consistent selection pressures, right, over maybe hundreds or thousands of generations, if you think about selective sweeps or background selection. So I want to talk about a model that's much more dynamic, right? Fluctuating selection. So fluctuating selection is defined as a shift in the magnitude or direction of selection over time. And we have a lot of evidence about this type of selection from phenotypes, right? So there's a lot of phenotypes fluctuate over time in a way that's too strong to be consistent with genetic drift. Classical study is a study in the scarlet tiger moth by Fisher and Ford in 1947, where there's this fluctuation in wing patterns. And then there's also a lot of theoretical studies that have been developed or that came out. But I think it's fair to say that kind of the whole landscape or the picture has changed dramatically with the study by Bergland et al. In 2014, where they monitor a Drosophila population in Pennsylvania. They look at population genetic data over time, and they see the strong variation, fluctuation in the allele frequency at around 1,700 loci. So there's a statistically significant fluctuations in allele frequency in this population, right, over 3 years. So you see this allele frequencies that go point isn't here. Okay, so that go up and down from spring to fall, and go down from spring to fall, and up from fall to spring, and so on. And there were some discussions about the statistical aspects of this study, but then also there were a lot of confirmations of this general pattern in Mercado et al. In 2021. They found similar patterns in European populations of Drosophila. In Behrman et al. 22, they found fluctuating selection with this kind of rapid adaptation to different seasons on the phenotypic level. And then there are also other approaches with cages where they kind of restrict or like stop gene flow, because gene flow could also be, fluctuating gene flow could also lead to similar patterns. So there's a lot of evidence in Drosophila that that's actually a real thing, but not only Drosophila. So we also see very recent studies in the non-binding midge, where we see like fluctuating selection, temperature adaptations on hundreds or thousands of loci. We see similar adaptation to temporal changes in salinity in the 3-spine stickleback, to changes in salinity in the water. There's a study using ancient DNA in horses, where we see that's a single locus that fluctuates over time. It's related to coat pattern of the horses. Then there's a study in Arabidopsis thaliana as well, adaptation again at the single locus to fluctuations in the soil salinity. And there are more recent studies that I didn't put up here in Daphnia and monkey flower, where there's also like fluctuating selection at multiple loci in the genome. So I think it's fair to say that there's more and more evidence suggesting that there is actually fluctuating selection on a very short time scale in a lot of species, and it also affects, at least in insect species, it affects a large proportion of the genome, right? Given that, so we were interested in what does this multi-locus type of fluctuating selection model, right? We observed those fluctuating alleles over maybe at most 3 years in those studies. What are the consequences if that continues to happen in terms of genome evolution? And we were particularly interested in the effect on genetic diversity and closely related effective population size. So we tried to study this idea, like approach this idea by simulating fluctuating selection, multi-locus fluctuating selection. We used the segregation lift model by Whitman et al. 2017. So that's a model that leads to this fluctuating alleles at many loci. And there's a whole distribution of amplitudes under this model. So we simulated under this model, and we found that there's a single parameter, the epistasis parameter in this model, that kind of modulates the average amplitude of the fluctuating alleles. So what we did basically is we estimated this epistasis parameter. We estimated an epistasis parameter that fits most closely to the empirical data sets, particularly the one by Bergland in Pennsylvania and Mercado in Europe. So in this case, for example, right, you would predict an epistasis parameter that's somewhere in the range of 4 to 12. So we simulate these fluctuating alleles. If you're more interested in models of the mechanistic models of fluctuating selection that can explain those like hundreds of loci that are fluctuating, we just recently published a review on fluctuating selection, so we talk a bit more about that in this review. But we also spent a lot of time like optimizing the simulation framework. So there's mostly based on SLIM and MS Prime. So we really tried to optimize computationally those simulations. So from a practical point of view, if you want to run simulations of kind of non-standard models of selection, have a look at that paper. It might be useful. But I won't go into in a lot of detail, but what we simulate is the chromosomes 2 and 3 of Drosophila using this empirically estimated recombination map here. So the red line is the recombination map that we simulate under. We introduce fluctuating variants, so variants that are directly under selection, randomly in the genome. And then we also introduce neutral variants. So we use those neutral variants to estimate the variance effective population size. So we measure the change in the allele frequency of those neutral variants over a year, and then the variance of those, that allele frequency change allows us to estimate the effective population size. We also introduce neutral variants in 3R in part of the chromosome 3, where we didn't introduce fluctuating variants just to see if there's any difference. That's more like a sanity check, so that we wouldn't expect the variance effective population size to be different in kind of linked regions versus unlinked regions. But so if you do that, what we see is that, so the red line is, those are neutral simulations, right, where we estimate variance effective population size in simulations where we didn't introduce those fluctuating variants. And we basically estimate the census population size, the population size that we simulate under. Those are really large simulations. We really simulate like under 1 million flies here. However, as soon as we introduce fluctuating variants, right, the effective population size quite drastically reduces, about 50%. It's a factor of 2 reduction in effective population size. And similar patterns can be seen in unlinked regions. So another thing that we also saw is that the equilibrium effective population size is reached fairly quickly after introducing those selected variants, where we introduced the selected variants in the beginning at 50%, but some of the smaller effect fluctuating loci get lost or fixed fairly quickly after their introduction. Okay, so now those simulations suggest there can be a quite strong effect, but it simulates under a specific number of selected sites and a specific mean amplitude, right? In that case, it's 154 selected sites with a mean amplitude of 3.7%. But it's actually fairly hard to estimate the actual number of selected loci from empirical data. You know, it's like based on how conservative, how strict you want to be with the definition of selected loci from empirical data, you can come up with like maybe on the order of a few hundred loci all the way down to maybe just 9 loci. So it's just very hard. We don't have good information about the number of selected loci because those data sets obviously are a mixture of directly selected loci and also linked neutral variants that also fluctuate. So what we did is we simulated under a range of different numbers of selected loci and mean amplitude, and we get very different results, right? If there's a lot of directly selected variants in the genome that fluctuate with a fairly large amplitude, maybe on average 6.7% here, then you get a fairly large effect. If you have just 9 loci, but they fluctuate quite strongly with 13.6% on average, then you get still a decent effect. Right? But if you have just a small number of loci with very subtle fluctuations, then you get a very small effect, which obviously is a big problem because we don't know the actual number of directly selected loci in the genome. So we wonder what can we do about that, right? What can we actually measure? So a very naive approach was then what if we just know the most strongly fluctuating locus and we can measure its amplitude fairly accurately, right? Can that kind of predict the effect of fluctuating selection on effective population size? And it turns out it's actually a very good predictor. So if we just plot under those models, under those different simulations, just one allele that has the largest amplitude, right, on the x-axis, and the relative reduction in effective population size on the y-axis, we see a fairly linear relationship. So if you take the largest amplitude from the Bergland and Mercado studies, both agree on that fairly closely. It's about 40%. We get a reduction in effective population size of about 50%. In conclusion, fluctuating selection can lead to a strong reduction in effective population size. In our simulations, up to 75%. Now, the most strongly fluctuating alleles might allow us to very effectively predict the reduction in effective population size, right? We don't need to know this number of the total number of causal loci. And this suggests a reduction of 50% in empirical data. And the application of this type of model to other species will hopefully enhance our understanding of fluctuating selection and its impact on effective population size across different species, and maybe also contribute to this discrepancy kind of between effective population size and census population size, right? That's particularly large in insect populations. So that's work by grad student and now postdoc in my lab, Olivia Johnson, mainly, also contributions by Josh Schmidt, Ray Tobler, and Mike Wittmann. I just want to advertise really quickly evolutionary genetics position at the assistant or associate professor position at Penn State. So it's a really collaborative, very supportive community here at Penn State in the biology department. So yeah, please contact me if you're interested in such a position. Okay, yeah, I want to thank you for listening and happy to take questions. Yeah.
Nice talk. So in your simulations, is the population size
it's constant. Yeah, it's soft selection.
Sweeps and background selection.
Yeah, yeah. Mm-hmm. Yeah.
Does fluctuating selection always increase affected population size? Could there be scenarios where actually it increases affected?
Yeah, there's very, very nice work by Michael Wittmann, just came out recently. It's just from a theory paper where she shows it on a genome-wide level, it almost always reduces effective population size, right? It increases diversity very closely linked to the selected sites, kind of like balancing selection, but genome-wide overall it reduces diversity. Yeah.
Would generation time impact like how much reduction we see in fluctuation time? Or I'm sorry, how fluctuating selection impacts the reduction in any? Like if there's a shorter generation time, then
Yeah, kind of. We didn't test that, but obviously it depends on the scale of the environmental fluctuations versus generation time. Our simulations right now are very focused on Drosophila because that's where we have most of the evidence. Yep.
I think I'm beginning to talk, but could you define what statistical distribution you use to develop fluctuation?
Yeah, we take the.
Do you vary the correlation properties or the time scale?
We sample the effect sizes from certain distributions, so to get the distribution of fluctuating variance. So it's based on a model by Wittmann et al. Yeah. So there's some distributions that we sample from. Thank you.
All right, good morning. I'm Kerry Cobb. I'm a postdoc at Mississippi State with Dr. Megan Smith. I'm here today to tell you about how we think we can use machine learning to overcome some problems with detecting introgression. So introgression is what we call the process that results in genetic variation from one divergent lineage being transferred into another divergent lineage through hybridization, where importantly some of that persists and could be detected in samples taken from a lineage. And we have a lot of methods that can detect this, and they work quite well, but they make some maybe unrealistic assumptions about our data. So one way that we can detect introgression would be to use a convolutional neural network. These form the foundation of a lot of the image recognition algorithms that are out there. This is a supervised method, so it requires that our data have labels, so we need to know if there is or is not introgression, and we need a lot of data. So a real data set like that doesn't exist. So what we do is simulate it, and then for input into the networks that I'm going to show you today, we're using a joint site frequency spectrum. If that's not familiar to you, what's important to know is they contain a signal of the evolutionary history of the two lineages, and we can treat them like we would an image for a CNN. The way a CNN works is it takes a filter and slides it across the data, and it extracts local features from the data that can then be used as input into a neural network, which can do classification and tell us is there or is there not introgression in the data. So I trained a network like this using a data set simulated under a range of realistic parameters. In half of the data sets, there was introgression, or there was not introgression, and in half of the data sets simulated migration during a period of migration after secondary contact. And then I used test data simulated under the same model to see how well this network does at classifying these data. And it worked really well. The network was able to perfectly identify whether there was or wasn't introgression in 1,000 test data sets. But as I mentioned, the methods we have for detecting introgression, they make some assumptions about the data. We assume that the processes in our model are the only processes that are generating the patterns of diversity that we observe. And when this is violated, we get model misspecification, and that can have a negative impact on the performance of our methods. One example of model misspecification is ghost introgression. Ghost introgression is introgression coming from a lineage that is not the focus of our analysis, coming into one of the lineages that is. This could be because of a limitation of the method, like with these statistics, or because we're unable to sample it. maybe it's extinct, maybe a freezer malfunctioned. Regardless of why, it's a problem because it produces patterns of genetic variation that our methods might mistake for introgression between the lineages that we're focused on. So to demonstrate this, I simulated a new test data set under a model very similar to the model that I used to train, except that one lineage received a pulse of introgression from a ghost lineage. And I used these data then to test the effectiveness of that previous network that was trained with the simpler model to see how well it did at classifying these misspecified data. And in this case, it did pretty poorly. We see quite a few false positives relative to false negatives, and it's misclassifying a large portion of our data sets that did not have introgression here. Another way to look at this is with these ROC curves. They show the false positive rate and the true positive rate at different classification thresholds. But what's important to know is that a completely random classifier would have a line that looks like the gray dotted line here, and a perfect classifier would have a line that looks like this straight line across the top. And we can get an aggregate measure of the performance across all the classification thresholds with the area under the curve, the AUC. And ideally, we want an AUC close of 1, or as close to 1 as we can get. And so each line in this plot is a different training and testing replicate performed with the same training and test data. The blue lines represent the tests where there was no misspecification that did really well. The orange lines represent the tests where there was model misspecification. And we can see that these orange lines are far from optimal, and there's a lot of variation among replicates. And then the AUC is only 0.8. So there's an example of how ghost introgression negatively impacts a test for introgression. This is not a problem just with machine learning. Other methods have the same problem. And this is also not limited to ghost introgression. There's many other processes that can confound these tests. So what can we do about it? Well, it would be great if we could just model all of these other processes, but that's just a non-starter with a lot of methods. It's not a great solution for machine learning. As we add more dimensions to the model, we need to increase the number of simulated data sets to sample all the parameter space. And that could quickly get out of hand, and that'll greatly increase the computation time and the resources we need. And then even if we tried that, there's things we aren't going to account for because we don't know the true history of our empirical data. So an exciting possibility is to apply something called domain adaptation. So in domain adaptation, the goal is to find features in the data that are shared across 2 domains. So the first domain is the source domain. This is the domain of our initial training data that you saw today. In our case, the data simulated under a simple model. The second domain is the domain of our empirical data. As you saw before, if we try to classify, if we try to use a classifier trained on the source domain, try to use that classifier on the target domain, we will misclassify a lot of our samples, as you can see there in that cartoon. So the process of domain adaptation, it takes data from the target domain, and it tries to identify features that are shared by the 2, and then ignores the rest, ignores those features that might be confounding the training or the test. So one very appealing aspect of domain adaptation is that we can potentially use empirical data sets for training because we don't need as much data from the target domain as we need for the source domain when we're training. And we also don't need labels for the target domain. So that's an exciting possibility that might be available to us with this approach. So what does this actually look like in practice? Well, domain adaptation encompasses a variety of different methods, but some that have proven to be really effective in a lot of applications are based on the use of some kind of adversarial network. Generally speaking, these networks take data from from the source domain and from the target domain, and they identify the shared features in those data by having two components that are in opposition to each other. So we have this classification component of the network that's trying to classify the data as, in this case, introgression or no introgression. And then there's this second component of the network that is trying to discriminate between source data and target data. And by the end of training, what we want is to maximize the classification accuracy of the network and minimize the discrimination accuracy. So we used a network like this, specifically one known as a CDAN, and we trained and tested a model using the simple two-lineage model as our source domain data, and then the slightly more complex ghost introgression model for the target domain. And then we tested this network using data from the target domain simulations. So here were the results from the original trained network without domain adaptation that were quite bad. And with domain adaptation, we see a huge improvement. This classifier is almost perfect, and there's a lot more consistency among replicates. We have an AUC of 0.99 after using domain adaptation. We can do this kind of cool visualization of how well the domain adaptation is working to identify the shared features from these two domains by plotting the encoded features with a t-SNE. So a t-SNE is just, it's a lot like a PCA. It's a dimensionality reduction technique. What's being plotted are features extracted by the convolutional filter in the CNN from the trained network. And so without domain adaptation, we can see a lot of separation here in the encoded features between the source and the target domain. And after domain adaptation, we see this really nice overlap between the two, which makes sense because it was performing much better at classifying our data. Oops. Okay. So we found that training replicates often fail to produce an effective network. These networks are a little harder to train than your standard ones, but fortunately it's fairly easy to recognize when this happens by looking at the accuracy across the training epochs. So in order to produce a network with high test accuracy, it's necessary for the discrimination accuracy that you see in orange here to initially reach a high level and then drop off and remain low around 0.5. The one on the left there is an example of one that performs well, and the one on the right is one that performs bad. It just rises and kind of stays there. Sometimes they just stay flat the whole time. But what's nice about this is we've been exploring the use of some like objective criteria that we could use to assess training and like a priori before we even test these networks. And then we could even stop it early to save time during training, which is nice. So in summary, model misspecification negatively affects tests for introgression. A lot of processes can generate false positives. That's nothing new, but we think that machine learning with domain adaptation is a promising solution to this. It allows us to allows the network to learn features that are shared between the target and the source domain, ignore the features that might be confounding our test, and then what's really cool is we might be able to use empirical data to train these. We're exploring that right now to see if the really remarkable results that we're seeing now with the simulated data can translate to empirical data. But this fact that we can use these empirical data to aid our training means we could potentially overcome like almost any type of model misspecification as long as there are features shared between our source and target domains. And then finally, something that's kind of interesting is we found that there's maybe a nice objective way to evaluate training of these networks. Finally, want to mention that I used MS Prime to do all the simulations and TensorFlow to train and test the networks. We used a package called a Python package called Adapt to do the domain adaptation, and we owe a lot of gratitude to Professor Jean-Francois Gu at MS State. He, on top of everything he's doing, does an excellent job maintaining our computer cluster, which we rely on heavily for this kind of work. So, and then thank you for coming. Have a little time for questions. Yeah.
Have you tested how well your machine learning algorithms work when you test models that are outside of like even the domain expansion? Like test a model with like two ghost lineages and see if it still works.
No, no, we haven't. Well, we're working on, right now we're trying to use empirical data. So that would certainly have processes in addition to the simple Gaussian regression that we've tested with. But we haven't done any, the point of the domain adaptation is to adapt using data that has all of the unmodeled processes that could exist. So, but no, we haven't done that. Thank you.
Okay, good morning. I'm the Abby that Lindy mentioned earlier, but I'm not going to be talking about DFEs now. Instead, I'm going to be introducing a new permutation approach for detecting natural selection in allele frequency time series. And this work was done as part of my undergraduate thesis. So I'll get right into it. Whole genome allele frequency time series provide a very detailed window into evolutionary processes, especially when compared to data from just a single time point. However, inferring selection from this type of data is still quite difficult, partly due to genetic drift, but also because selection itself is stochastic. Previous methods have often estimated selection for single trajectories, but this requires selection to be quite strong in order to detect a signature. And they also often rely on temporally consistent selection, pushing the trajectories in one direction throughout time. But we know that selection can often be much more complicated than this. And so if we neglect the possibility that it may push trajectories in different directions throughout time, we may underestimate its total effect on a time series. And so more recent previous work has instead focused on analyzing relationships between trajectories in a time series. In particular, Buffalo and Coop found positive covariances between allele frequency time changes throughout time, and so that suggests linked selection. And while this is more statistically powerful, it also relies on temporally consistent selection. And using summary statistics like this means that a lot of the information available isn't fully utilized. And so we wanted to develop a method that used information about each trajectory's behavior and also accounted for the possibility that selection may switch direction and strength throughout time. And so our approach essentially builds on a permutation test. We permute trajectories with respect to time and look for how their characteristics change. So we can describe a trajectory by its initial frequency and then a series of increments. And under neutrality, the evolution of a trajectory should be statistically symmetrical. And so those increments can be reordered and interchanged without affecting the properties of the trajectory. Now, this isn't the case if natural selection is involved because of its temporal structure. And so if we permute those increments, the initial and final frequencies remain the same, but the interior path changes. And so we remove any temporal structure. So we can apply this to each trajectory in a time series, and if there are discrepancies in properties from before and after permutation, that suggests that there was some sort of temporal structure in the original time series. And so in particular, we're most interested in how the maximum deviation of a trajectory changes, or how far it travels from its initial frequency. So for trajectories with positive autocorrelation, or trajectories that tend to move in one direction consistently throughout time, those tend to see a decrease in their maximum deviation. And alternatively, for trajectories with negative autocorrelation, or ones that tend to switch direction often and bounce around, those tend to see an increase in their maximum deviation. And so this is an example of what permutation does to a simulated autocorrelated time series. If we look on the left, there's little to no visual change in the trajectories themselves. But if we look at the distribution of all of the maximum deviations, we see a drastic shift. And so the direction and magnitude of that shift is going to depend on the sign and strength of the autocorrelation present in the time series. So under neutrality, or when there's no autocorrelation or selection happening, we find that there is no change to the distribution, and we've used the Kolmogorov-Smirnov test to confirm this. And this is to be expected, since the original time series would have been random and statistically symmetrical anyways. And just like the single trajectory cases, if we have a time series with positive autocorrelation, we see a shift towards lower maximum deviation values. And if we have a time series with negative autocorrelation, we see a shift towards higher maximum deviation values. And so we found that that shift in either direction, depending on the sign, is amplified as the strength of autocorrelation increases. So in principle, we think we should also see these changes in empirical data. And so that's our main goal, is to apply this permutation approach and detect temporal structure in empirical genome-wide allele frequencies. But before we can do that, and before we can represent autocorrelation, we need a stochastic model for the selection coefficients of the trajectories, because just using genetic drift wouldn't have any temporal structure. So we've been using a first-degree autoregressive model for the selection coefficients. Essentially, for a given allele, its current strength of selection depends on some portion of its most previous value plus some noise, so denoted as epsilon here. And this is the simplest way and easiest way to introduce autocorrelation and a temporal structure, but it could be extended by including more terms that depend on selection further back in time. So we could have a selection coefficient that depends on its 10 most previous values instead. And when we simulate trajectories using this and the Wright-Fisher model, there's 3 parameters that we've been tuning. Alpha will control the strength and sign of the autocorrelation. Sigma is the standard deviation of the noise term in the selection coefficients. So we've been focusing on normally distributed noise. And N is our effective population size. So that'll control the strength of genetic drift. And by changing these 3 parameters, we'll observe different levels of change to our maximum deviation distribution. So to quantify that shift in the distribution, we've been looking at the bin-wise difference in the various maximum deviation values. And so this results in a curve that characterizes both the direction and the magnitude of the shift we see. And so if we can get this curve for some goal distribution with unknown parameters, like from a data set, we can simulate using various parameter values and compare the resulting curve to our goal curve. And we can treat that as an objective function. So we can try to minimize the mean squared error between the two and try and get them to match. So by doing that, we'll have simulated a time series that shows similar behavior to the one in question, and hopefully that gives some insight into the underlying selection structure. So we haven't yet constructed a likelihood function for our parameters, so we've been conducting global searches over ranges of parameter values. And this objective function is variable because we can only simulate a finite number of trajectories. But given simulation results, we found that this is working reasonably well. If we look at results from attempting to fit simulated data with positive autocorrelation, we can see 2 regions with relatively low mean squared error. And some parameter values can be kind of ruled out and were unlikely to cause the observed shift from the simulated data. So for the region in the bottom right, parameters in that area have very high stochasticity in the selection coefficients. So while parameters in that area will cause a similar shift to the maximum deviation distribution, the distribution itself looks wrong, the shape isn't quite right. So even without knowing the true parameters, we can kind of rule that region out based on shape alone. And similarly, if we try to fit simulated data with negative autocorrelation, we again see two regions of low mean squared error. This time, the region with high stochasticity and the selection coefficients is in the top right. So again, here, we would see similar changes to the distributions, but can kind of rule this region out based on just the shape of the distributions alone. So in either case, we can narrow it down to one region of plausible parameters. And so based on these results, we think this should also hold with empirical data. If we apply our permutation procedure to the Drosophila dataset from Bargy et al., we found that each of the 10 replicate populations shows a shift towards higher maximum deviation values. And so that suggests negative autocorrelation. Now the Drosophila were left to evolve in stable lab conditions. And so if selection really is pushing the trajectories in different directions, even in a time-independent environment, this is a pretty surprising result. But before we can attempt to fit this, we have to consider how alleles that are being fixed or lost in the population are affecting our procedure. So everything I've discussed so far has assumed that not many of the trajectories will hit the boundaries of 0 and 1. And so we've shown that our process should work for data sets when that's the case. Unfortunately, with the Bargy data set, around 50% of the trajectories are either fixed or lost in the population. And for trajectories that hit the boundaries, their maximum deviation won't change after permutation. And so while it's tempting to discard and ignore them and only focus on trajectories that will show a change in behavior, we think this may be emphasizing trajectories that are less likely to hit the edges and therefore more likely to switch direction. So it may be enhancing negative autocorrelation. So we're still thinking about how to go about fitting and modeling data sets that have this type of behavior because it gets a little bit more complicated. But to sum it up, we found that permuting time series removes temporal structure and reveals useful signatures of selection. In particular, the changes to the maximum deviations of the trajectories give an estimate to the sign and strength of autocorrelation. Going forward, we also want to look at local versus global genome analysis. Right now, we've been assuming one alpha value for the entire time series, but we know that certain areas of the genome may experience linked selection. And so it may be the case that using different alpha values for different sets of nearby alleles may be more appropriate. Like I said earlier, we can also extend our autoregressive model to a higher degree and include more influence from previous selection values. But our main goal is to be able to apply this to empirical data sets. So right now, we are working to fine-tune our procedure to make it more robust to fixation and loss. So I'll leave it at that. And I'd just like to thank my supervisor, Dr. Jason Bertram, for all of his help on this project, and also to NSERC for helping to fund our research. And if there are any questions. Yeah.
Great talk. This might be a bit extreme, but I was wondering, so this is assuming that the allele frequencies are known without error, and so those are like, estimates of like, you have individuals that you could permute individuals across time and maybe look at directly the uncertainty in estimating those allele frequencies. Is that something that might be worth considering, or is that?
It may be in the future. Right now, we were focused on a very broad, general approach to this. So looking just at like genome-wide selection and being able to detect something there, but I'm sure it could also be narrowed down to more specific individual cases in the future once we fine-tune things a bit. Yeah, of course. Yep. Whoever.
Some practical questions. Does it change anything if we don't have same space between time point, like between time point 1 and 2, 5 generations and 3 generations and 10?
Yeah, so we've been exploring, in empirical data, there often is like, it's not every generation that they sequence. So we've been exploring also using coarse graining. So for our simulations, we can run it for a set number of generations, but only look at every 10th generation or every 5th generation. So that's something we are exploring is different generation lengths between sample points.
With sequencing sampling, we accept that there is a noise around the frequency. If you
Not yet. So the empirical work is still very fresh, and we're still working towards that. So we haven't yet considered the sampling noise and how we're going to work with that. We just really want to get a handle on the intuition behind what this does, and just know that in theory and in principle, it should work. Okay, thank you. Okay, thank you.
Hi everyone, thanks for coming today. So my name is Viv, I'm a postdoc in the Jensen lab at ASU, and this rather verbose title really is just a two-step approach for demographic and DFE inference in coding sparse genomes. So quantifying the relative importance of selective and neutral processes in shaping genetic variation has long been a focal point in population genetics, and two fundamental entities in this regard are the demographic history of a population, which determines the historic and ongoing efficacy of genetic drift, and the distribution of fitness effects, which has already been talked about a lot today, which determines the selective effects of new mutations, whether they be deleterious, effectively neutral, or beneficial. So it's not surprising then that a great deal of effort and research has gone into attempting to infer these sorts of models. And an important genomic signature in this regard is in the site frequency spectrum, which is the distribution of allele frequencies within a population. And we have three site frequency spectra plotted at the bottom here. So in blue we have what would be our neutral expectation. So this is a population of constant size that's neutrally evolving. And then we have two deviations from this neutral expectation. So in orange we have a population that's expanding, it's growing in size, that's also neutrally evolving. And then in green we have a population that's of constant size but is under purifying and background selection. So that's the purging of deleterious variation and linked neutral variation. And two things are really evident here. Firstly, we can see that these deviations from neutrality leave a signature in the site frequency spectrum. We have this leftward skew towards rare alleles. And secondly, that both selective and neutral processes can leave similar signatures, which can, of course, result in misinference if they're not accounted for. So really our question here is, if population size change and selection can both leave the same sort of signatures, then how can we infer the underlying population history and the DFE without each being confounded? So demographic inference being confounded by selection and DFE inference being confounded by population size change. So probably the most commonly used approach for DFE inference is the two-step approach of Keatley-Walker. So in step one, demographic inference is performed on the synonymous site frequency spectrum, and a demographic model is inferred with a single step size change in population size and the time of that change. And then conditional on that demographic model, in step two, DFE inference is performed on the non-synonymous site frequency spectrum. Now the main assumption with such an approach is that synonymous sites are neutrally evolving, and so background selection isn't accounted for. If synonymous sites are linked to deleterious non-synonymous sites, they're going to be under background selection. That's going to affect demographic inference in step one, and of course, because step two is conditional on that demographic model inferred in step one, it's also going to bias DFE inference. And so this is something that has to be accounted for when using such an approach. An alternative approach is that introduced by Jerry et al. In 2020. This is a joint approximate Bayesian computation inference approach. Here the demographic history and the DFE are jointly inferred, and so because you're jointly inferring the parameters, you're avoiding the biasing effects of selection and population history. And because you're using the approximate Bayesian computation framework, you can harness multiple aspects of the data, not just the site frequency spectrum, but also information on divergence, linkage to equilibrium, population structure. Of course, the downside to such an approach is it's going to be computationally intensive because you're jointly inferring the parameters of both the demographic model and the DFE. You're exploring a very large parameter space. So thus far, it's only really been used to infer single step size changes in demography. And so with both these approaches, we see that we're really looking at quite simplistic demographic models. And of course, we know populations have really undergone a much more complex demographic history. And so here what we're proposing is a two-step approach that's applicable in organisms with coding sparse genomes. And so in step one, we identify non-functional regions that are distant enough away from functional regions that we can be sure that they're not undergoing background selection. And with region-specific mutation recombination rates and information on population structure, we can perform demographic inference on these regions using, for example, coalescent or diffusion approximation approaches. Or if we want to harness those multiple aspects of the data, we can use an approximate Bayesian computation approach. And because we're not worried about DFE parameters at this point, we can infer a far more complex model with a greater number of parameters. Then in step two, conditional on the demographic history inferred in step one, we can identify functional regions, again with region-specific mutation recombination rates. And in an approximate Bayesian computation framework, we can infer a discrete distribution of fitness effects, where we're inferring the proportion of neutral, weakly, moderately, and strongly deleterious mutations. And of course, if one wanted to add further categories for example, weakly beneficial mutations, then of course you could. So we applied this approach to human data. And so our first step here is to curate a set of non-functional regions. We identified regions that are at least 10 kilobases away from any functional region. Now, we know this is far enough from a functional region to avoid the biasing effects of background selection based on the analytical calculations of Jerry et al. In 2020. And with region-specific rates, we have a total of 146 regions for demographic inference. And so what we're doing is we're running simulations. So we're simulating our demographic model under a range of parameters for each of our regions, comparing our simulated and empirical data. We ran coalescent simulations in MS Prime. And because we're working with human data here, and this is really an example application, what we're doing is we're just optimizing parameters from the extensive literature in humans as opposed to actually developing a demographic model from scratch. And so what we did is we took the demographic model from the Gutekunst et al. Paper in 2009, expanding it slightly, so we also include the South Asian population. So we sampled 4 populations in total, the African, European, East Asian, and South Asian populations. And we also modeled the more recent Bantu expansion in the African population. Down the bottom here, we just have a table with a list of parameters, and the papers from which we've drawn those parameter ranges. And so we have our tau parameters, which are the split times of populations. The B parameters are our bottleneck parameters, so the severity of the bottleneck when a population is dispersing. And the R parameters are our growth rates per generation. We also have migration parameters, of course, between the populations, but we sample from an entire range of 4 NM between 0 and 10. And in this top right, we can see the parameter ranges themselves. So on our x-axis, we have the parameter in question. The blue bars are the entire parameter range, and the orange data points are our best fitting parameters. And you can see the demographic model with those simulated under those best fitting parameters below that. The most notable difference here between the Gutekunst and Gravel best fitting parameters are we have a much larger final African population size, and that will be because we're modeling that Bantu expansion. And then on the left, we can see the fit of a number of summary statistics between our empirical and our simulated data for each of our populations. So we have a number of segregating sites, Tajima's D, which measures the skew in the site frequency spectrum, mean R squared, which is a measure of linkage disequilibrium, and FST, which is a measure of population structure. And all of those are for each of our 4 populations except FST, which is going to be between populations, of course. The data points are for the mean value across our simulated regions, and then we've got the variance on those, which are the confidence intervals. And we can see we've got a pretty good fit across the board, across our populations and our summary statistics, suggesting that our demographic model under these parameters fits the empirical data well, putting us in a position to move on to step 2 and DFE inference. So again, we have to identify a number of regions for DFE inference. Our criteria here is making sure that these regions are distant enough from other functional regions to ensure no interference between them. Now, Jerry et al. In 2023 performed the joint inference approach I mentioned earlier. They performed that in humans, so they'd already curated a set of regions. So we're using the same regions as they are here, again with region-specific mutation and recombination rates. So this time, because we're simulating selection, we're simulating forward in time in SLIM. And we simulated under the distribution of fitness effects from that Jerry et al. Paper, which we can see in the bottom right corner here. So on the x-axis we have F0, which is the proportion of effectively neutral mutations, and then Fs1 to 3 are the proportion of weakly, moderately, and strongly deleterious mutations, respectively. And considering they've accounted for the effects of background selection and of population size change by using the joint inference approach, and we're using the same set of curated regions as they are, we'd expect this DFE to fit our data well. And you can see that once again we have a pretty good fit here. We're also looking at divergence here, which is of course going to be an important consideration when we're modelling selection. And we can go on from here and then look at how much, if we add a beneficial DFE category, what proportion of beneficial mutations we can incorporate into this model while still maintaining a good fit to the data. And so just to conclude, we've really shown the viability of such an approach in coding sparse genomes here, and that's going to be the key criterion here. If you don't have these coding sparse genomes, you're not going to have access to these long non-functional regions that aren't going to be affected by background selection, and therefore you'd have your inference biased in step 1 with this approach. But when we do have access to these non-functional regions that are unaffected by selection at linked sites, this approach would be viable, so for example in other primate species. And once you have such an evolutionarily appropriate baseline model, and you also factor in mutation recombination rate heterogeneity, then you're in a position where you can start inferring more episodic processes, such as selective sweeps and balancing selection. And because you've accounted for those constantly occurring processes, you're going to have a lower false positive rate with that sort of inference. Okay, and with that, I'd just like to thank my PI, Jeff Jensen. This work was funded by his NIH grant, and thank members of the Jensen and Pfeiffer labs. And the Jensen lab is currently recruiting postdocs, so if anyone is interested, I can tell you more about it. Thank you. Any questions? Perfect. Thank you.